our cancer or neoplasm studies. Cancer can be defined as a mass of tissues formed as a result of abnormal, excessive, uncoordinated, autonomous, purposeless proliferation of the cell. Actually, this proliferation of these cells are not purposeful, and it is uncoordinated, and it is autonomous as well as abnormal and excessive. It is a disease characterized by abnormal and uncontrolled cell division attacking the surrounding tissue and organs. So, when the cells grow uncontrolled, it attacks the surrounding tissue and organs and also the distant body parts. It can affect also the distant body parts because of the circulating blood and lymph. It circulates with the blood and lymph. The cells circulates with the blood and lymph. Neoplasm is or tumor is an abnormal mass of tissue. It is an abnormal mass of tissue. It results either from uncontrolled division or from obstruction of cells. Then this may be of two types, as we have already seen: benign tumor and malignant tumor. Benign tumor is non-cancerous cellular growth, and it does not invade the nearby tissues or spread to other body parts. It will not spread to other body parts, and it is not invading the nearby tissue. At the same time, malignant tumor, it is a cancerous, undefined and unpredictable cellular growth which invades the nearby tissues directly by the lymphatic system or circulatory system. So this malignant tumor, it is unpredictable cellular growth, undefined cellular growth, and it invades the nearby tissues directly by the lymphatic system or circulatory system. Okay, and it is also cancerous. Next, here we can see the normal cells and cancerous cells. In cancerous cells, there are many cells that continue to grow and divide, and there will be variations in size and shapes of the cells. And it is having a nucleus which is larger and darker than normal. There will be abnormal number of chromosomes arranged in a disorganized fashion in cancerous cells. And also there will be cluster of cells without any boundary. But in normal cells, there will be a boundary. Then we can see the difference between benign tumors and malignant tumors. Benign tumors develop slowly in a localized manner. Abnormal cellular growth pushes the nearby normal tissues. So it grows by pushing out the normal tissues. It does not invade the nearby tissues and also does not spread to other body parts via blood and lymphatic channels. This benign tumors is not spreading to other body parts via blood and lymphatic channels. It can be surgically removed because it is localized. And regarding malignant tumors, comprises of an unclear mass of cells growing very rapidly, and it is not localized. It lacks local growth, invades the surrounding tissues. This cancer cell or malignant tumors invades the surrounding tissues and reaches the other body parts with the help of vascular and lymphatic channels. This cannot be removed as it spreads widely throughout the body and thus leading to death. This cancer can lead to, malignant tumors can lead to death. And here also we can see the difference between benign tumor and malignant tumor. In benign tumor, cells are not cancerous and won't spread. Regarding malignant tumor, cells are cancerous and can spread to other tissues and organs. Now, neoplastic cells can be characterized with the help of following that distinguish them from normal cells. So here we can see the difference of normal cells and neoplastic cells. Regarding neoplastic cells, uncontrolled proliferation. The proliferation of cancer cells are not regulated by Regulatory process of tissue growth. Usually we have a regulatory process in our body regarding tissue growth, but in the case of cancer there will be uncontrolled proliferation and loss of function, invasiveness, it invades, rapid spread of cancer cell to secondary site, to another site it will be spreading, it will be invading another site. Then metastasis, that is the fourth character of this neoplastic cells, that is a migration of primary tumors to another site through blood vessels or lymphatic system, resulting in formation of secondary tumor known as metastasis. So metastasis is the formation of secondary tumor by migrating. The primary tumors will be migrating to another site through blood vessels or lymphatic system. The mechanism of carcinogenesis. Cancer cell develop from normal cell in a complex processes consisting following 4 phases of cell transformation. There are 4 phases of cell transformation in mechanism of carcinogenesis. Phase 1, that is the initiation phase, single or multiple exposure of carcinogenesis. Then change in genetic material of normal cell and alteration in DNA structure. In phase 1, which is the initiation phase, there will be single or multiple exposure of carcinogenesis, change in genetic material of normal cell and alteration in DNA structure. In phase 2, that is the promotion phase, ability of carcinogenesis to induce proliferation in initiation tissue and stimulate tumor formation. So in phase 2, there will be stimulation of tumor formation. That is the ability of carcinogenesis to induce proliferation in initiated tissue. There will be a proliferation in the initiated tissue. In phase 3, it is a conversion phase. Normal initiated precancerous cell become cancerous cell. Here, normal initiated precancerous cell becomes a cancerous cell. In the phase 3, it is a conversion phase of normal cell to precancerous, normal cell to cancerous cell, and alteration in genetic material also takes place. And phase 4, it is a progression phase. It is a spread of tumor cell from a primary lesion to a distant site called metastasis. It is the metastasis phase. It is a progression phase. It is spread of tumor cell from primary lesion to distant site. It is having 3 steps: attachments, degradation, and locomotion. Attachment means this cancerous cell attaches to the extracellular matrix. During degradation phase, local degradation of extracellular matrix. When cancerous cell attaches to the extracellular matrix in attachment phase, during degradation phase, that extracellular matrix becomes degraded. Then locomotion phase, that is a cancerous cell locomote to the secondary site through the locally degraded matrix. So already degraded matrix is present, so cancer cell locomotes to the secondary site through this locally degraded matrix. Next, classification of cancer. We have already studied classification. First one is carcinoma, that is the surface cancer, that is glandular or from parenchymal epithelium. So cancer arises from the surface region, that is a glandular or parenchymal epithelium. An example is cancer of breast, prostate, lung. Next is sarcoma, cancer derived from connective tissue. Example, fibrosarcoma, it is fibroblast malignancy, and myosarcoma, that is muscle cell malignancy. So for carcinoma, cancer of breast, prostate, lung are the examples. For sarcoma, fibrosarcoma, it is fibroblast malignancy, or myosarcoma, that is a muscle cell malignancy is the example. And next is the lymphoma or leukemia. It is derived from hematopoietic, that is the blood cancer. Leukemia is the blood cancer. It is derived from hematopoietic blood forming cells and develops from the precursors of the WBCs. In leukemia, cells multiply abnormally, diffuse within the bone marrow. It is from the bone marrow. It starts from the bone marrow, diffuse within the bone marrow to multiply further and replace the normal blood forming cells. So here The normal blood forming cells will be replaced by cancer cells. Thus, neoplastic cells spill over into the circulation and large number of abnormal cells circulate in the peripheral blood. Thus, these cancer cells circulate in the peripheral blood. Next, etiology or causes of cancer. This also we have already seen: tobacco, smoking, then alcohol consumption, genetic problems, excessive sunlight exposure, especially during these days. UV radiation is more in the sunlight, so excessive sunlight exposure. Then obesity, viruses, some viruses also form cancer. Then failure of immune system or defense mechanism. Immune system failure also causes cancer. These are the etiological factors, that is gene and chromosomal abnormalities. This normal cell will be affected by virus, gene and chromosomal abnormalities, then hereditary factors or other factors. Other factors include toxic stimuli, carcinogens, physical agents, chemical agents, etc. These normal cells become abnormal cells. So there will be failure of immune system or defense mechanism, which leads to abnormal proliferation of cells, which leads to malignancy or cancer. General etiology and pathogenesis of cancer. Before going to the pathophysiology part, what is the difference between pathogenesis and pathophysiology? Pathogenesis describes the origin and step-by-step development of a disease. So here pathogenesis describes origin and step-by-step development of cancer, how it starts and progresses. But pathophysiology focuses on the disordered physiological mechanisms and functional changes in the body as a result of that disease. Here we will see the general etiology and pathogenesis. There is much difference in pathogenesis and pathophysiology. Pathogenesis being the step-by-step development and its origin, but pathophysiology is a disordered physiological mechanism and functional changes occurring in the body as a result of that disease. So here we can see the normal cell and acquired DNA damaging agents like chemicals. These are the etiological factors: chemicals, radiation, viruses, etc. And normal cell, sometimes the DNA gets damaged, but usually there is a successful DNA repair and which again becomes a normal cell. But sometimes there is a failure of DNA repair. That may be due to inherited mutations in genes affecting DNA repair or genes affecting cell growth or apoptosis. This results in the mutations in the genome of somatic cells. So it causes activation of growth promoting oncogenes or alterations of genes that regulate apoptosis or inactivation of cancer suppressor genes. This altogether causes expression of altered gene products and loss of regulatory gene products, and due to clonal expansion, additional mutations or progression of mutations occurs, and which leads to heterogeneity and malignant, which produces ultimately malignant neoplasm. This is the general etiology and pathogenesis. Now we will see detailed etiological factors. It is etiological agents that induce cancer. One, as we have seen, is environment factors. It is tobacco, smokes, then diets, environment pollutants, etc. Then heavy smoking causes lung, oral cavity and esophagus cancer. Heavy smoking causes lung, oral cavity and esophagus cancer. And excessive intake of alcohol causes liver cancer. So due to smoking, lung, oral cavity and esophagus cancer, but excessive intake of alcohol causes liver cancer. Then chemical carcinogens are there, like nickel compounds, cadmium, arsenic, nitrosamines, trichloroethylene, arylamines, benzopyrene, aflatoxins, reactive oxygen radicals, etc. These are the chemical carcinogens due to food or due to pest, pesticides. Those who are working in pest control areas, they will be affected by some of the pesticides. So nickel compounds, cadmium, arsenic, nitrosamines, trichloroethylene, arylamines, benzopyrenes, aflatoxins, reactive oxygen radicals, etc. Due to our food also, mostly these vegetables, fruits, etc. will be exposed to these pesticides, excessive pesticide, so it may affect. And also physical carcinogen, UV rays, as we have seen, excessive standing in the sun, exposure to sun, that is UV rays, in sun rays there will be UV radiations, the ionizing radiation, X-rays and gamma rays. Those who are working in X-ray department, etc., they have to be careful, and also gamma rays. Then biological carcinogens are there. It is virus, bacteria, etc. Virus example, Rose sarcoma virus, that is RSV. It is the first discovered retrovirus causing cancer. Retrovirus causing cancer. Then these viruses are known as oncovirus. It is oncologist is oncology. No? So cancer causing viruses are known as oncovirus. Example, human papillomavirus, HPV, then Epstein-Barr virus, that is EBV, hepatitis B virus, and herpes virus, then hepatitis B and C viruses usually related with hepatocellular carcinoma, that is liver cell carcinoma. The cytomegalovirus associated with the Kaposi sarcoma. Human papillomavirus is the chief suspect of cervix cancer. Then there is one bacteria, that is Helicobacter pylori, which is present in the GIT. Usually it causes diarrhea, which sometimes it results in cancer. Then endogenous factors are there, that is mutations, that is inside our body. Mutations, change in DNA replication, metabolic reactions generating reactive oxygen species. There are metabolic reactions which generates reactive oxygen radicals, and immune system defects, aging, etc. These are the endogenous factors. Now we are coming to pathophysiology. In pathophysiology, mutation inactivates tumor suppressor gene. Due to mutation, tumor suppressor gene will be inactivated. Then cell proliferation results in cell proliferation, which again results in mutation inactivates DNA repair genes. Mutation also inactivates DNA repair genes, which results in again mutation of proto-oncogene, which creates an oncogene. Then mutation in proto-oncogene creates an oncogene. Then mutation inactivates several more tumor suppressor genes as it progresses. As mutation progresses, it inactivates several more tumor suppressor genes, and it ultimately results in cancer. The factors or agents which are responsible for change of normal cell into cancer cells are known as carcinogens. So carcinogens are agents or factors responsible for change of normal cell into cancer cell. All cells carry certain cancer-producing oncogenes. In our body, all cells carry certain cancer-producing oncogenes. These oncogenes are genes that are responsible for induction of tumors. Under certain conditions, these genes are triggered to multiply rapidly into malignant neoplasm. This is the abnormal growth of tissue. So, there are genes inside our body which are responsible for induction of tumors. Under certain conditions, these genes will be triggered to multiply, and they become malignant neoplasm, which is an abnormal growth of tissue. Now, cancer pathophysiology. Common pathophysiological process of malignant tumors or cancer development in the organism is damage to the genetic apparatus of cells. This damage includes mutation, disturbance of gene expression, activation of tumor promoter gene, inactivation of tumor suppressor gene. Here there will be activation of tumor promoter gene and inactivation of tumor suppressor gene in our body. There will be mutation, disturbance of gene expression, then activation of tumor promoter gene and inactivation of tumor suppressor gene. The damage to the genetic apparatus of the cell along with inactivation of anti-tumor genes. So there will be damage to the genetic apparatus of the cell along with inactivation of anti-tumor genes takes place both together. These are essential for the development of malignant tumors. This results in development of malignant tumors. This inactivation of tumor suppressor gene is one of the natural physiological reactions of the organism. In every organism there will be tumor suppressor gene, but inactivation is one of the natural physiological reactions of the organism. In the organism there will be inactivation of tumor suppressor gene, and when this reaction becomes a pathophysiological condition of the organism, it results in cancer development. When the inactivation of tumor suppressor gene becomes pathophysiological condition of the organism, it results in cancer development. This is a multi-step process involving mutation and selection for cells with progressively increasing capacity for proliferation, survival, invasion and metastasis. So this will be a multi-step process which involves mutation, selection for cell with progressively increasing capacity for proliferation. These cells will be having increasing capacity for proliferation, survival, invasion and metastasis. So this is a multi-step process. It is involving mutation as well as selection of cells with progressively increasing capacity for proliferation, survival, invasion and metastasis. That's all about the pathophysiological part. Now we are coming to the growth phase, which involves S phase, G2 phase, G1 phase, etc. In S phase, each of the 46 chromosomes is duplicated by the cell. G2 phase, the cell double checks the duplicated chromosomes for error, making any needed repairs. Then mitosis and cytokinesis, then G1 stage, cellular contents excluding the chromosomes are duplicated. All cellular contents excluding the chromosomes are duplicated in G1 phase, and G0 phase is the cell cycle arrest. This is the growth phase of cancer cell division, which we will see in detail. G0 phase. This phase is termed as the resting phase or G0 alpha phase. This is also known as resting phase or G0 alpha phase. In this phase, cell cycle arrest and halt to proliferation. In this phase, cell cycle get arrested and halt to proliferation. Due to resting condition, this phase is resistant to various chemotherapeutic agents. So in this phase, none of the chemotherapeutic agents will be applicable or effective. In G1 phase, this phase is also termed as the early protein synthesis phase. This is the early protein synthesis phase. In this phase, cell synthesizes proteins, RNA and DNA molecules. In this phase, cell synthesizes proteins, RNA and DNA molecules. Next is the S phase. This phase is also termed as synthesis phase. S phase is also known as synthesis phase. In this phase, cell synthesizes a DNA protein with the help of DNA polymerase. There are some enzymes here. DNA protein synthesizes a DNA protein with the help of DNA polymerase, RNA polymerase 2 and topoisomerase 1 and 2 active enzyme. These are the enzymes involved in which cell synthesizes DNA protein. So the enzymes involved are DNA polymerase, RNA polymerase 2 and topoisomerase 1 and 2 active enzyme. Next is the G2 phase. This phase is also termed as pre-mitosis phase. This is a pre-mitosis phase, the phase which is just before the mitosis phase. In this phase, various cellular and structural components involved in mitosis for synthesis. At the end of this phase, the number of chromosomes is doubled. Here, in G2 phase, number of chromosomes is doubled, and the cell becomes ready for active division. When the chromosomes are doubled, it is ready for active division. That is the M phase. This phase is also termed as mitosis phase. This is the shortest phase of cell cycle. M phase is the shortest phase, that is a mitosis phase. It is further divided into prophase, metaphase, anaphase and telophase. It is again divided into 4 phases: prophase, metaphase, anaphase and telophase. 2 daughter cells are formed after this phase. There will be 2 daughter cells after this. Mutation results in loss of control of the cell cycle, differentiation and cell-to-cell adhesion and interaction. Here, mutation results in loss of control of cell cycle, then differentiation and cell-to-cell adhesion and interaction. These changes cause morphological changes in the cell or tissues. So this cause morphological or changes in the appearance in the cell or tissue. And these are the signs and symptoms: chills, loss of appetite, fatigue, fever, weight loss, anemia, shortness of breath, abdominal pain, chest pain, cough, heaviness. So these signs and symptoms may depend upon what type of cancer that person is having. Sometimes chills, loss of appetite, fatigue, fever, weight loss, anemia, shortness of breath, abdominal pain, chest pain, cough, heaviness. So this weight loss, anemia, etc. may be due to fever, etc. may be due to leukemia. Then abdominal pain may be due to GIT cancer, or cough may be due to lung cancer, like that. These are some of the key signs and symptoms of cancer. So, very heavy night sweats, then fatigue, unexplained bleeding or bruising, unexplained pain or ache, unexplained weight loss, unusual lump or swelling anywhere on your body, a new mole or changes to a mole, skin changes or a sore that won't heal. So very heavy night sweats, fatigue, unexplained bleeding or bruising, unexplained pain or ache, then unexplained weight loss, then unusual lump or swelling anywhere on your body, a new mole or changes to a mole, the skin changes or a sore that won't heal, that will never heal, then croaky voice, hoarseness or a cough that won't go away, mouth or tongue ulcer that lasts longer than 3 weeks, coughing up blood, difficulty swallowing, breathlessness, persistent heartburn or indigestion, unusual changes to the size, shape or feel of breast due to breast cancer or chest, including nipple or skin changes, persistent bloating in the stomach, appetite loss, change in bowel habit such as constipation, looser poo or pooing more often, blood in your poo, etc. Then unexpected vaginal bleeding, including after sex, between periods or after the menopause, blood in your pee, then problems in pee, croaky voice, hoarseness may be due to the throat cancer, then mouth or tongue ulcer due to mouth cancer, and coughing up blood may be due to lung cancer, difficulty swallowing, esophagus cancer, breathlessness again due to lung cancer, persistent heartburn or indigestion, GI cancer, unusual changes to the size, shape or feel of breast cancer, persistent bloating, appetite loss, then change in bowel habit such as constipation, loose poo, etc. due to GI cancer, unexpected vaginal bleeding, maybe due to cancer in the vagina, then including after sex, between periods or after the menopause, blood in your pee, problems in pee, etc. are the signs and symptoms of cancer. Next, we are going to see the complications of cancer. Local effects like compression, obstruction, infiltration. Then systemic effects like hormonal, nutrition, immunologic and idiopathic effects may be there. Treatment-related effects. Cytotoxic chemotherapy. Due to chemotherapy, there are many adverse effects. Bystander effects caused by death of tumor cells. Then toxic effects of radiation therapy. These are the complications. So local effects, compression, obstruction, infiltration, systemic effects, hormonal, nutrition, immunologic, idiopathic. The treatment-related effects, there are many adverse effects, cytotoxic chemotherapy, then bystander effects caused by death of tumor cells, toxic effects of radiation therapy. The diagnosis can be diagnosed by screening, that is types of cancer, cancer of breast, cervix, colon and rectum, and prostate cancer. Cancers of breast we can screen by mammography and self-examination. We can self-examine. Then cancers of cervix by pap smear, cancers of colon and rectum by rectal examination, blood test, colonoscopy. Then prostate cancer by prostate specific antigen and transrectal ultrasonography. Then tissue biopsy, usually they do first examination will be tissue biopsy, that is procedure of removal of a tissue specimen for microscopic studies. That means wherever there is doubt there is a cancer, suspect there is a cancer, they remove a tissue from that portion and removal of tissue specimen for microscopic studies and study it microscopically. Then CT scan is there, MRI scan will be there. The laboratory methods are there, involve cytological, histological and gene profiling methods for diagnosing cancers. Blood test also is there for tumor markers. And prevention and treatment. Prevention methods are there and also treatment methods are there. Prevention by avoiding risk factors like tobacco, overweight, alcohol, and sexually transmitted disease. We have already seen that is etiological factors, there is tobacco, alcohol, etc. So we can avoid taking tobacco, then or alcohol, then we can control our overweight, then sexually transmitted disease also we can control. And dietary emphasis on vegetables, fruits, whole grains and fishes. By diet we can control, we can prevent, that is emphasis on vegetables, fruits, whole grains and fishes. Then surgery, we can remove it by surgery. Then radiation therapy is there, chemotherapy is there. And medications like NSAID, that is symptomatic treatment for pain, we give NSAIDs, and vitamin D, folic acid. And vaccinations are hepatitis B vaccine is there. Now we'll see which uh, chemotherapeutic agent is effective in which phase. Like there's in anaphase, antibiotics, example bleomycin, actinomycin, etc. will be effective. And in GI phase, also these are effective. This GI phase, G1 phase. G1 phase is the early protein synthesis phase. Then alkylating agents like nitrogen mustard, melphalan, chlorambucil, cyclophosphamide, etc. will be effective in metaphase. In metaphase, plant alkaloids like vinblastin, vincristin, etc. also will be effective. The antimetabolites like methotrexate, 5-fluorouracil, 6-mercaptopurine, etc. are effective in S phase, that is a DNA synthesis phase. And these alkylating agents, nitrogen mustard, melphalan, chlorambucil, cyclophosphamide, etc. are also effective in S phase. Thank you.